So here I want to look at a basic introduction to cell membranes, but I'm actually considering kind of deeper concepts, but providing the explanation in a simple way. So, concepts. In terms of the cell membrane, we all know that the cell membrane is composed of a phospholipid bilayer, and this phospholipid bilayer is what makes the border between what's extracellular and what's intracellular, right? And essentially, we have these hydrophilic heads facing away from the center, and hydrophilic heads, and we have these hydrophobic tails which face hydrophobic tails which face inward, and this is all to do with polarity as well. So we've talked about the phospholipid bilayer, and this phospholipid bilayer, each of these phospholipid molecules are actually moving and rotating and moving sideways and rotating. So because of the lateral movement as well as the rotation, that all allows for a mobile lipid bilayer. Okay, it's mobile. That means it's moving, it's fluid. Essentially, this is all composed of fluid, isn't it? So it's mobile, it's moving. Let's look at the components. Now, in terms of, you've got the phospholipid bilayer, that just means the phospholipid molecule, phosphate and lipids, essentially. And you've got glycolipids as well. You've also got glycoproteins. So you've got different components of the bilayer. That's absolutely fine. It's mobile. There is movement. Now, because this is a phospholipid bilayer, what kind of substances do you expect to move past and move through intracellularly? You expect lipid soluble substances, and for larger sort of protein molecules or ions, charged substances, small charged substances can pass easily, but lipid soluble molecules the easiest. But for extra substances, you will need special gateways, special channels, special proteins to allow for that transfer to occur. Now, the fact that this is a mobile lipid bilayer allows for a number of processes. Here's a process that's happening over here, and this process, here's exocytosis and here's endocytosis. So our lipid bilayer is this very dynamic, cool structure, and it also allows for things to occur such as endocytosis and exocytosis. But exactly how do endocytosis and exocytosis occur? They occur through receptors. Now, I want to talk a little bit about the concept behind receptors. Then I want to talk a little bit about the concept behind fluidity, which I already kind of addressed. Receptors allow for endocytosis and exocytosis to occur. Endocytosis and exocytosis is for sort of larger molecules or substances that can't pass easily through the lipid bilayer. So that's what receptors are for. What exactly is a receptor? By the way to remember the sort of hydrophobic heads and hydrophobic tails, when somebody's scared, a cat's scared, for example, it kind of hides behind its tail, or a dog, okay? And that's how you remember that something that's hydrophobic or scared is like that. Now, receptors are basically for substances that cannot just go intracellularly easily. That's what receptors are for. Otherwise, they can just flow straight in through the lipid bilayer without a care, okay? Now, these receptors, what are receptors essentially? Receptors are when you have, let's say, with one hand, you're catching a ball, and at the same time, as soon as you catch that ball with your other hand, let's say you start waving at somebody. Receptors work kind of like that. On the cell surface membrane, they are going to catch substances, but let's say that's a receptor, but it's actually going to alter or have modifications in the biological system further along. So, the attachment of a certain substrate to a receptor causes biological modification to the system further along the receptor, okay? It's not very difficult. Just imagine catching a ball and then doing something else with your other hand, or waving or something. That's what a receptor is. It doesn't have modification right at the surface, okay? And like we said, this was endocytosis, and endocytosis was the process of having a substance and pinching it in, and the cell membrane you need it to be fluid and mobile and dynamic for it to be able to enclose a substance and take it in. Exocytosis is kind of like the opposite of it. This is exocytosis, sorry, this is endocytosis. Endocytosis is like you take the substance, you enclose it, and you pinch off and fuse the membrane, and it bring it back. Let's talk about fluidity. Now, fluidity is kind of defined as the dynamic movement of the lipid bilayer and this fluidity is impacted by, let's say, temperature. A higher temperature will allow for more fluidity of the lipid bilayer, okay? Now, another thing that we can talk about now is our friend cholesterol. Let's talk about it over here. Here it is. These are little structures that are present in between the phospholipid bilayer, and that is cholesterol. Cholesterol is another lipid, and it looks like this. It doesn't actually look like this. It looks like this. Now, these cholesterol molecules, what do you think they will do to fluidity? Will they increase or decrease fluidity of the phospholipids, let's say? They will decrease fluidity. Why? They will decrease fluidity because just imagine, it's very logical. You've got these phospholipid molecules. In between, you stick like a bit of random pieces of cholesterol. That's going to limit the lateral movement of the phospholipids, right? Yes, but there's an exception. Some of the cholesterols have kinks in them, okay? And these kinks mean, let's say if somebody has sort of a broken leg, they're kind of unable to walk. So these are unsaturated cholesterols. And the saturated, or you can remember saturated for straight. And these are straight cholesterols, okay? So when they have a kink, so unsaturated, this is the exception, unsaturated cholesterol is going to increase fluidity. And that's because when there's a kink, there's still room for more movement as compared to with packing lots of straight cholesterols in between. I think this is kink, so it's unsaturated. Okay, last thing I want to talk about is lipid rafts. What's a lipid raft? A lipid raft is where you have, essentially, it's a dynamic, mobile cluster of phospholipids with other sort of functional molecules such as sphingolipids and cholesterol. Okay, so lipid raft, cluster of highly dense phospholipids with functional lipids, which are sphingolipids and cholesterol. And these kind of just float around. They float around within the phospholipid membrane, the bilayer, and these float around and they're really, really good. But I don't know, they just float around and they're dynamic and they're mobile, and these kind of things help us in processes such as endocytosis and exocytosis.